Hello everyone, my name is Fiona, and this summer I've had the pleasure of working at Dr. Shisiru's lab to investigate the role of RAC1 on iris activity in Coxsackievirus B3. So let's get started. So I looked at CV-B3 because it could result in mild flu-like symptoms, but it could also result in severe myocarditis in young children. And as of now, therapies are very inadequate. So finding treatment and prevention is extremely important. And this is exactly what Dr. Shisiru's lab has been doing, which is to find enteroviruses tissue tropism to see what types of tissues or cells CVB3 could replicate in. And so I looked at the internal ribosome entry site, which is found within the 5' untranslated region of RNA. And this promotes the internal initiation of protein synthesis using cap-independent mechanisms. So, short to say is that it's very essential for viral protein synthesis or viral infection. And then I looked at RAC1, which is a small GTPase that is crucial for regulating cellular processes. And the reason is because that previous research on RNA pull-down assays using Drosophila brain extracts and proteomic analysis discovered that RAC1 could be a potential host factor. So RAC1's regulation of iris activity could also be tissue specific. So I based off my research on this question, which is, could RAC1 affect the iris activity of CB3? So my methods, I used SF268 cells, which represents like neuronal cells, and RD cells representing muscle cells. And then I conducted transfection. So I did knockdown and overexpression for both cell types. So for knockdown, I would have two types, which is two samples, which is negative control siRNA or RAC1 targeting siRNA. And for overexpression, there are three. So either the empty vector control, or the wild-type RAC1, or the RAC1, which is the N17 RAC1. And I would then transfect all of my samples with CV-B3 IRES reporter RNA for luciferase assay to later look at the luciferase activity. And I did triplicate studies for most of the experiments. So for knockdown, it's very according to what I predicted, which is there is a significant decrease in iris activity of over 50%. And for RD, there was not a significant difference between when RAC1 was knocked down or when there was no knockdown. And this is right because this is predicted because it's tissue, RAC1's regulation of iris activity is tissue specific. However, for overexpression, SF268, there was a significant increase between vector control group and the wild type form. There was not a significant increase between vector and N17 form, but that could be because my vector samples were duplicates while my N17 samples were triplicates. So it required a really different numbers for it to be statistically significant. So this part was understandable. However, for the RD samples, overexpression samples, it's really contrary to original hypothesis. I expected there to not be a significant change between vector and RAC1 overexpression. However, as you can see, there was a lot of statistical significance. So ideally, I would conduct this experiment again to confirm the results, considering all the relationships are statistically significant, but I ran out of time, so these are the results I have now. But ideally, I would conduct this experiment again. But yeah, thank you so much for your time, and I also want to thank Dr. Shixing and Zhong Han on their guidance and opportunity of leading me through this project. Thank you. Bye.